Bismillahirrahmanirrahim. In the name of Allah, most gracious, the merciful. I am Dr. Muhammad Farhan Nasir from University of Education, Lahore, Pakistan. The topic of my research is effect of organic and inorganic arsenic oxide on hematological parameters of freshwater air-breathing fish, Sirhinus mirgala from South Punjab, Pakistan. The present study aimed to assess the survival response in terms of hematological changes of Sirhinus mirgala exposed to arsenic and arsenic trioxide in laboratory condition. In this investigation, the oxidative biomarkers in the blood sample of freshwater fish Sirhinus mirgala exposed at sublethal level of arsenic. Arsenic is included in the naturally occurring element. It is existed all over the world and more in the natural environment. Inorganic arsenic is easily absorbed in man by the oral route. In humans receiving orally less than 10 mg travelent inorganic arsenic, the absorption rate was found to exceed 80%. Arsenic occurs in the environment in inorganic and organic form. Arsenic is existed in the trivalent form, which is more toxicity than the pentavalent form of arsenic. The trivalent form of arsenic is more soluble in aquatic environment. Many such compounds like arsenic produce in the environment. Environmental toxicity in these environmental toxins can induce physiological and biochemical changes in fish and lead to inhibition of growth and death in case of acute poisoning. Many such compounds like arsenic produce the environmental toxicity and bioaccumulation of arsenic in the blood and the body of fish may cause the infertility in the fishes. It may also cause the liver damage and cell death of fish. The freshwater of fish, Sirhinus mirgala, was collected from the ponds located in Muzaffargarh and Dera Ghazi Khan region of South Punjab, Pakistan. The size range of fish samples was studied from 10 gram to 500 gram with length of 15 centimeter to 25 centimeter. The fishes were acclimatized with 1% KMNO4 to remove all pathogens from the surface of fish body. The fishes were maintained in the aquaria at a room temperature 28 degrees centigrade and the fish were introduced into 10-liter capacity containers of water containing specific metal arsenic. After the recovery period, all such treated fish were separated from the experimental container and blood samples were collected from 3 experimental individuals from each group and each time with an interval of 24 hours during the experiment. Blood of the fish was collected by puncturing the blood of fish and collected in the EDTA vial and the samples were analyzed by the hemocytometer to get the values of red blood cells, white blood cells, hemoglobin, and other blood parameters. Results: chemical situation of the experimental tank. The temperature was maintained between 22 to 29 degrees centigrade. The light period, there was 11 hours of day and 13 hours of night. Alkalinity was 150.6 to 176.8 mg per liter. And the water hardness was maintained between 176 to 202. Chemical properties of arsenic and hydroxide. The melting point of arsenic is 36 and its density is 5.7, while arsenic trioxide, its boiling temperature is 869 Fahrenheit, while melting is 594 Fahrenheit, while arsenic pentoxide, its boiling temperature is 315 degrees centigrade and its density is 4.32 gram per centimeter cube. Different hematological components under variable sublethal concentration of arsenic in Sirhinus mirgala. The total RBC in control group in the first 10 days it was 3.56, at 20th day it was 3.48, and at 30th day it was 3.52. While with 10% SLC it was 3.11, 2.72, and 2.14. While with 30% SLC, it was 2.51, 1.95, and 1.52. If we talk about the white blood cells in control group, at 10th day, it was 10.56. At 20th day, it was 10.37. At 30th day, 10.52. While with 10%, it was 12.61, 14.52, and 16.37. While with 30%, it was 13.66, 14.95, and at 30th day, 17.82. And if we talk about the hemoglobin in control group, it was 8.51, 8.61, and 8.56. While with 10%, it was 6.79 at 10th day, 6.14 at 20th day, and 5.31 at 30th day. while with 30% SLC, it was 4.18 at 10th day, 3.21 at 20th day, and 2.64 at 30th day. The WBC count of Sir Highness Minigala was increased with the increasing concentration of arsenic, and the increase in the number of WBC was found to be 29.36 to 44.17 and 69.39% at 10, 20, and 30 a day exposure at 30% sublethal concentrations, respectively. The hemoglobin content of Sir Highness Minigala in 10 and 30% sublethal concentration showed decreasing trend with a significant reduction of 20.21, 28.69, 37.97, and 50.88, and 62.71 and 69.16 at 10th, 20, and 30 a day of exposure, respectively. Here in the figure, you can see the exposure period with 10th day, 20th day, and the 30th day. The mortality percentage with the increased concentration of arsenic Sir Highness Minigala, as you can also observe that as the concentration of the arsenic was increased, the mortality rate was also increased. So it means it have a very lethal effect over the growth of the fish as well. Continuous exposure of lethal elements cause the increase of red blood cells in the fish and then sudden decrease in RBCs. With the decrease in RBC, the HP level of the blood was also decreased. Multiple forms of hemoglobin allows fish to adjust more efficiently to physiological stress such as varying water temperature and oxygen concentration. The increased concentration of white blood cells in this study was because of having direct activation of the defense system of the fish by having under the influence of any toxic element. Leukocyte is directly proportional to severity of stress condition in maturing fish and is a result of direct stimulation of immunological defense due to presence of heavy metals in wastewater pond. This is correlation with the report of cerevidin in the freshwater fish. The increased concentration of white blood cells in this study was because of having direct activation of the defense system of the fish by having under the influence of any toxic element. Leukocytosis is directly proportional to severity of stress condition in maturing fish and is a result of direct stimulation of immunological defense due to the presence of heavy metals in wastewater pond. This is in correlation with the report by Saravanan in freshwater fish. The toxicity of arsenic on the hematological parameters such as hemoglobin, RBCs, and WBC were significantly decreased because the arsenic affect the animal and led to anemia. It has also reported that pollutants generally cause change in the hematological parameters of the fish. Iqbal et al. Have also observed that reduced level of RBC, HP, and PCV in the Sir Highness Minigala on the fish treated with lead, the MCH and MCHC increase in the blood of Sir Highness Minigala exposed to nickel. The observed amount of MCH and MCHC indicate the hydrochronic microcytic anemia. The reduction in RBC count and HP content after 30 days of exposure indicates anemia associated with erythropenia. Our results are similar to the earlier reports of these scientists. Sir Highness Minigala exposed to arsenic concentration showed fall in number of lymphocytes due to decrease in non-specific immunity of fish. Leukocytes are involved in the regulation of immunological function in many organisms, and increase in the white blood cells in stress animals indicate a protective response to stress. The decrease in white blood cells count during acute and sublethal treatment may be due to toxic effects in head kidney of fish being primary site of hematopoiesis. Blood parameters were not affected when adult lake whitefish exposed to 0, 1, 10, and 100 microgram of arsenic gram per food. Low levels of arsenic toxicity fishes cause reduction in red and white blood cells. It is also reported a decreased level of hemoglobin and packed cell volume, that is PCV, in Clarice petricus exposed to arsenic. Sir Highness Minigala, when exposed to arsenic, showed decrease in granulocytes, erythrocytes, hemoglobin, hematocrit values. Contamination of water bodies by arsenic and its compound is big threat to all the aquatic organisms, especially fishes. They enter inside the body where skin, gills, gastrointestinal tract, and accumulated in vital organs, and once carried by the blood, Aquatic organisms accumulate arsenic mainly as inorganic form, but few organisms like bacteria, phytoplankton, etc. transform inorganic arsenic species into organic forms. Once toxic elements like arsenic may enter inside the body via food and drinking water, they produce many deleterious effects on organisms. Government of different countries should monitor anthropogenic sources which are contaminate aquatic body and increase public awareness. Thank you so much.